And our first of three speakers is Alexander Yuile, who is a medical oncologist and PhD candidate and a member of the brain cancer group based at Royal North Shore Hospital in Sydney. And Alex is going to be talking with us about the link between PSMA and VEGF looking for predictors of response to bevacizumab. And I'm sure he will explain those acronyms to us all. Over to you, Alex.
So, thank you, Hariana. So, I'm, my name's Alex. I'm a medical oncologist and a PhD student at Royal North Shore and at the Colling. So I'll be talking about a slightly different angle, but it's the same aims in terms of supporting people with brain tumours and supportive care with that. So the angle that I'll be looking at will be looking more at biomarkers. So what is a biomarker? So I've got a picture of a crystal ball here because if I had one, or if we had one, it would make everybody's lives a lot easier and would be super helpful. So if anyone knows where I could get a crystal ball, please let me know. But in the meantime, we use these things called biomarkers. So they're the characteristics or something in the brain tumour that or to do with the person with the tumour that gives us an idea of what their outcomes might be, or how to treat them. And often there's a focus on treating in terms of outcome of survival and prognosis. But the angle I'm sort of presenting today is we're also looking at how to use these as markers to treatment that would improve people's quality of life and would more likely have improved symptom control with certain treatments. So what we've in particular looking at is the symptoms from the swelling of the tumour, which is extremely common. So in the first picture we've got here is a picture of the green blob is the tumour, and then you've got the outline of all the blood vessels, and you can see that the tumour is able to sequester and produce all this extra blood vessels to feed it and to help it grow, and particularly gliomas and particularly glioblastomas do that, and they do that through this marker called VEGF, but the problem is because they make all these messy multiple chaotic vessels, they're leaky. So we've got this bucket which is the blood vessel, but it's leaking fluid, not blood but fluid, and that causes a lot of swelling, which leads to a lot of the major issues that have that brain cancers can cause, the huge impact of quality of life, and a lot of these has been amazing to see everyone's work addressing all of this today, and a lot of these different things, seizures, behavioural changes, have already come up. So what we can do is block this formation with a treatment called Avastin, which is used a lot in Australia. That stops the messy blood vessels, it stops the leaking, and then that can help the symptoms. But the problem is, particularly in supportive care, is there's no perfect drug, and everything we do also can cause side effects. So what we are trying to do is find ways to predict, because not everyone responds to this. And so why aren't they responding? Can we improve the response to help symptoms? And can we pick out the people who are definitely going or are much more likely to respond? So this is where, unfortunately, it gets a little sciencey, but we've looked at different tumours, and we can see that what Avastin blocks, this VEGF, it increases with another marker called PSMA. And we can see that looking at the microscope slides, and that's what these pictures are showing. And what we are trying to do, and what we want to look into, is we have a PET scans or scans that can look at the PSMA marker, and we can see that. So we can see brain tumours that have that increased, and that we think that also means that the VEGF is increased. So we can pick who responds to bevacizumab, so we can help direct treatments, tailor it to the person, and hopefully potentially mean that we're decreasing the toxicity burden of treatment and maximizing response to improve quality of life. And the other thing that's quite exciting is that, or what we're looking into is the relationship between these two markets, because it's a relatively new concept about this relationship. And if we can flesh that out more, we might be able to leverage this association, so we can even help people who don't have symptom control with Avastin. Perhaps we can leverage this link to do something to help their control, and perhaps we can improve the response to people that do have symptom control with Avastin to make it longer and more durable. So that's sort of the snapshot of what I've been looking at. And I'd like to acknowledge my supervisors for my PhD, Dr. Helen Wheeler, Dr. Adrian Lee, Dr. Amanda Hudson, and Professor Michael Back and Professor Pavlakis, and the Brain Cancer Group and White Pearl Foundation, who fund all the research I've been involved in. I'm immensely thankful for, and of course the patients, consumers and their families and caregivers. And I'll leave you today with a picture of our daughter Mira, who turns 2 today. So I had to put that picture in somewhere. She loves meerkats, so thought that was an appropriate picture.